Hello, can you hear me? I'm Avery Selberg, and I'll be presenting on busted pH, which stands for the branch site unrestricted statistical test for episodic diversification, and it incorporates phenotypes. This is a collaboration between myself and Sergey Pond, as well as Maria Chikina and Nathan Clark. So the goal of, busted pH comes from the common goal in genomics, which is to understand the relationship between genotype and phenotype. We can do this by identifying selection on genes in a particular species or population with a specific phenotype, which is the genetic basis for morphological adaptation. We can use convergent traits, or traits that have evolved multiple times independently, to provide increased statistical power through their natural replications. In known case of convergent evolution, selection is repeatedly targeting the same gene. So to leverage this power of convergent evolution, there's been a widely used workflow that has been used in hundreds of papers. I'll go over kind of the gist of that. The first step is to choose a convergent species. In this presentation, I'll be talking about marine mammals. The next step is to perform a sequencing and alignment, or get this information from a database such as the UCSC 100-way alignment. So here we'll have a multiple sequence alignment of marine mammals and terrestrial mammals. The third step is to create a phylogenetic tree. I have here a species tree, but a gene tree is also often necessary. Here, you can't really see, but in boxes, we have the marine mammals, and those marine mammals are designated as the foreground species, whereas the remaining species, the terrestrial species, are designated as the background species. So this fourth step is the step that I'll be focusing on. This is the selection analysis. So in this step, we're looking to identify adaptive evolution in genes that are in the foreground species. So I'm a part of the HiFi lab. That's hypothesis testing using phylogenies. I'll be talking about ways that we can improve this step, but another commonly used method is PAML, which stands for phylogenetic analyses by maximum likelihood. So after the selection analysis is run, it's common to perform an enrichment analysis. This may be done using a program such as KEGG or Panther to identify like pathways or gene ontologies terms of those adaptively evolving genes. So now that we're focusing on these selection models, one alternative model is rate-based methods. To understand these rate-based methods, it's important to understand the difference between relaxation of selection and adaptive evolution. So I like to visualize this on a number line where we have the dotted line, we have neutral evolution, and to the left of the dotted line, we have negative selection or purifying selection. That's where DNDS, which is the non-synonymous to synonymous substitution rate ratio, is less than 1. And then to the right of the dotted line, we have adaptive evolution or positive selection. That's where DNDS is greater than 1. So if we start out with a gene that is under very strong negative selection, this gene is under evolutionary constraint. It's highly conserved. If over time the selective pressures on this gene change, so now that gene is under weaker negative selection, it's still under purifying selection, but it's not as strong as it was before. This shift signifies relaxation of selection. Now, that relaxation of selection is different than if that gene would suddenly become like adaptively evolving, because then the gene would be under positive selection. So rate-based methods such as RER-converged, which was developed by our collaborator, they identify genes at different rates, or the shifts in the selection values in the convergently evolving species. However, they cannot distinguish between the selective forces of relaxation of selection and adaptive evolution, because they both elevate mean evolutionary rates. So RER-converge is a great model. It was developed by our collaborators, but we wanted to have a way to identify adaptive evolution. So that brings us to codon models. Codon models inherently consider both the nucleotide and amino acid substitutions, and they do track the selective pressures as omega, which approximates DNDS, again, the non-synonymous to synonymous substitution rate ratio. So they do this by designating a foreground and background species in advance. So in the phylogenetic tree I have here, the foreground species are the marine mammals in orange, and the background species are the terrestrial mammals in blue. That way we can identify and compare selective pressures between these subsets of branches. And then codon models, they work as a finite state continuous time Markov process. So one existing codon model is PAML, or CODA-ML within PAML. But the problem with using PAML is that it uses very restrictive selective profiles that do not allow for positive selection in the background. So because of this, any selective forces in those background species, that's the terrestrial mammals in our example, they can be mistaken for selection in the foreground in the marine mammal species, which can cause uncontrolled rates of positive selection or of false positives. Now, there are some ways to rectify this. Our collaborators came up with a really nice way to do this, but it involves removing data, which can create biases and reduces power. And then additionally, this is not very well known. Many people just use CODA-ML as it is written, and they don't pay attention to selection in the background. I have here 4 papers that have done this, but there are hundreds more on Google Scholar that are doing this process. So to kind of attack this issue, we developed Busted pH, which again stands for the Branch Site Unrestricted Statistical Test for Episodic Diversification with Phenotype, and it tests for foreground-specific positive selection. Busted pH works in a series of 4 models and makes 3 comparisons between those 4 models. So the inputs of Busted pH include a species tree that denotes the foreground and background species, as well as a multiple sequence alignment. And from there, HiFi will create a gene tree for each individual gene that you run through Busted pH. So going to those 4 models, the first model is known as the universal alternative model. It gets compared to each of the 3 upcoming null models through a likelihood ratio test. So in this first universal alternative model, we are estimating omega or DNDS distributions for both the foreground and background branches. This is done from the data using maximum likelihood with a random effects likelihood model. So what I mean by distributions, by default, we have 3 omega values, which might represent positive selection, neutral selection, and negative selection. And so we have 3 values for the foreground species and corresponding percentages for the percentage of codon sites for each omega value that is being binned. So we have 1 distribution for the foreground here and 1 distribution for the background. Now, like I said, this model serves as the universal alternative for all the 3 upcoming models. So the first null model is used to test for foreground-specific positive selection or episodic diversifying selection. In this null model, we constrain the foreground species. So we say, like, what if positive selection was not allowed in the foreground by forcing omega to be less than or equal to 1. Now we do this because it serves as the null model, and then we can use the likelihood ratio test to compare this model to the unconstrained model from the previous step. Now conceptually, this model is very similar to what's done within PAML and CODA-ML and also previous HiFi models. Now where Busted pH becomes new is in these tests. This is the test for positive selection in the background species. So similar to the previous test, this time we're constraining the background. So we're saying, what if positive selection were not not allowed at all in the background. So we're enforcing omega less than or equal to 1 on all of the background branches. This allows us to use a likelihood ratio test to compare this null model to the unconstrained model from step 1. And like I said, this test is new to busted pH. The fourth and final model is to test whether or not the omega distributions are different between the foreground and background branches. So in this null model, we will have a single shared distribution over the entire phylogenetic tree. So we're kind of ignoring the foreground and background partitions. So once again, we're using this model and comparing it to the universal alternative model of step 1. And again, this test is a new addition to busted pH. So to help us interpret these 4 different models and comparisons, I have here a decision tree, which HiFi automatically runs through, but I have it illustrated here. So the first test is the test for positive selection in the foreground. Again, we have the unrestricted universal alternative model, which this time is compared to the constrained foreground model. If this test is not significant at a value of 0.05, then we can say there is no evidence of selection associated with the phenotype. If this test is significant, then we move on to the test for positive selection in the background. This time our null model was the constrained background model. If this test was not significant, then we move on to the test for the difference between the background and foreground branches. Once again, that test was where we constrain the foreground and background to a single omega distribution over the entire tree to serve as our null model. If that test is not significant, then we can say there is evidence of selection associated with the phenotype, but it is not statistically significant. There is not a statistically significant difference between the foreground and background selective regimes. If that test is significant, then we can say there is strong evidence of selection associated with the phenotype. So jumping back to that test for positive selection in the background, if that test is significant, then we look at the test for the difference between the background and foreground branches. If that test is not significant, then we can say selection is occurring with or without the phenotype, but with similar selective regimes. And if that test is significant, then we can say selection is occurring with or without the phenotype, but with different selective regimes. Now, I think this decision tree is a lot easier to understand if we look at empirical data. So I'll continue to talk about marine mammalian species. So going into this analysis, we had some observed morphological adaptations, which include limb reduction and thickening of the epidermis and some other adaptations that I have listed here. So our hypothesis was that genes related to these morphological traits of adapting to life underwater would be under positive selection at higher rates than other genes, specifically in the marine mammalian species, but not in the terrestrial mammalian species. So to do this, we used the data from the UCSC 100-way alignment, and we only pulled from the mammalian species. So the first gene that I'll be talking about that we looked at is COL9A2, which is a collagen hyaline joint cartilage protein. If we think back to our observed morphological adaptation of epidermis thickening in marine mammals, we thought that this gene might be under positive selection. And I have here the gene tree that HiFi produced for this specific gene. So looking at the data from running COL9A2 through busted pH, we have at the top of these circles here the largest omega values for the foreground, background, and the distribution over the entire tree. We'll notice that they're all greater than 1, which reminder that indicates positive selection. However, looking at the p-values, only the test for selection in the foreground, as well as the test for the difference in selection between the 2 sets of branches were significant. So in the test for selection in the foreground, our largest omega value was 5.7, and this value was the case for 0.5% of codon sites. That's in the COL9A2 gene in only the foreground species. And over the test over the entire tree, that distribution, the largest omega value was 15.48, which was the case for 0.33% of codon sites in the COL9A2 gene over the entire phylogenetic tree. So we can plug this data back into the decision tree. So first we have our test for selection in the foreground. This test was significant at a value of 0.05. Next, we have our test for positive selection in the background. This test was not significant. And then finally, we have our test for the difference between the background and foreground branches. And this test was also significant. So this tells us that there is strong evidence of selection associated with the phenotype in COL9A2, which agreed with our initial hypothesis there. So the next gene that I'll be talking about is this gene GSTM1, which is responsible for detoxification of ROS and environmental pollutants. Now, I'm highlighting this gene because of this paper I have listed down here from the Tien group. They found that, they studied the GST gene families, and they found that this gene in particular, the GSTM1, was under positive selection in pinnipeds, which includes seals and walruses. So I thought since we had this gene in our data set, I would look at these results specifically. So once again, all of these top values, the omega values, are greater than 1, indicating there was some positive selection. This time, when we did our test, the test for positive selection in the foreground and the test for positive selection in the background were both significant. However, the test for the difference in selection between the two sets of branches was not significant. So looking at our foreground distribution, our largest omega value bin was 18.19, which was the case for 1.3% of codon sites in the GSTM1 gene for the foreground marine mammal species only. And now for the background, our largest omega value was 2.56, which was the case for 26% of codon sites for the GSTM1 gene for the terrestrial mammal species. So when we look at the results put into this decision tree, first we're looking at the test for positive selection in the foreground. This did get a significant p-value. So next we have the test for positive selection in the background. This also achieved significance. And then finally we have the test for the difference between the background and foreground. This did not achieve significance. So based on this result through HiFi, we can say selection is occurring with or without the phenotype, but with similar selective regimes. And I wanted to put up here what HiFi prints out. They make it very clear. They say selection is acting on the branches with the phenotype and trait, but is also acting on the background branches. So this makes it very difficult to misinterpret the results we had here, unlike if you would use a different program such as CodeML. Okay, so Busted pH is available on our GitHub page, that top QR code, as well as I put some visualizations on the website Observable with that marine mammalian data set. I'd like to acknowledge the Pond Lab, specifically my advisor, Sergey Pond, as well as our collaborators, Nathan, Maria, and Amanda. Thank you.
Thank you, Avery. Are there questions to Avery? We have time for one question. Danny.
I don't have a question, but I really loved your decision trees.
Oh, thank you.
So then I get to ask a question. Can you go back to the example? I don't know if it's the last or the second last.
This one? GSTM1?
Yes, this one. I think this one. So here you don't see any difference between the two, but there's clearly the branches are very long, right? This on the foreground. So how do you explain this if it's if there's no difference there? Yeah.
So the branch length can account for both synonymous and non-synonymous change, but we're specifically looking for that non-synonymous change. So that might have some account for why the branch length was so long, would be my guess.